So welcome back, everyone. This is Dr. Balding. Right now, we're going to talk about how we know where genes are located in prokaryotes. How do we find the locus of a gene? Well, these days where we have the ability to sequence genes, but way before we had the ability to sequence a gene, we could actually determine where a gene was located on the bacterial chromosome. And it's kind of a cool set of experiments that I'm going to talk about right now. And I want you guys to understand some of the ideas that are behind this type of experimentation. So, whenever a bacterium that has a certain type of plasmid in it called fertility plasmid, and remember these plasmids contain extraneous, basically genetic information for bacterium. There's some bacteria that have plasmids, right? And some called fertility plasmids. Well, bacteria that have a fertility plasmid, so this is its chromosome and there's the bacteria, and let's just draw the plasmid with a little circle there. When they have a fertility plasmid, they're called F plus strains. And when they meet up with a bacterium that, say, has genomic information but no plasmid, that bacterium is called an F minus strain. All right? So, what the bacterium with the fertility plasmid can do is it has genes on that plasmid to synthesize a type of structure that we talked about before, a pilus. Okay, it's called a conjugation pilus or a sex pilus. All right, and that plasmid is replicated and transferred, right, to the recipient strain, the F minus strain, and then that strain too becomes an F plus strain. All right. So, but there are those situations where, on a rare occasion, let me erase that. I don't like the way I drew it. On a rare occasion, what can happen is there are bacteria that have a chromosome, right, in which this F plasmid has somehow integrated into the host chromosome. Okay? It doesn't happen that often, but when it does, we call those HFR strains. Okay, high frequency of recombination. And they behave just like F plus strains. So when they meet up with a bacterium that, say, doesn't have that genetic material, okay, this HFR strain can still form the sex pilus. And rather than just transferring the plasmid, what ends up in this host cell is a region, or the plasmid, right, sequence that was on the chromosome and part of the host chromosome that it takes with it. And it can take as much as the whole host chromosome with it into the new cell, right? Usually the sex pilus is broken somehow through movement and so forth of a culture or environment before this occurs. But if they're allowed to stay very, very still, the whole chromosome can be transferred into the recipient cell. Okay, so keeping that in mind, I'm going to erase all this. We go to this picture down here where you can see there is in this blender, and this is just representative of one event that's occurring, but there's a culture or a whole bunch of F plus strains, okay, that are actually HFR strains, right, that have this plasmid integrated into the host that have been mixed with F minus strains, okay, or an F minus strain, And we're looking to laterally transfer some genetic information from one of those strains to another. Okay? And so what happens is, let me add a page here. So I'll add a page. You can take, and I'll just do this for one gene. Let's just say we're looking at an enzyme that helps in making purines. You guys know what purines are? They're the nitrogenous base of one of of a couple of our nucleotides. Right? Let's just say we have a bacterial chromosome, and we have a gene for purine biosynthesis. Right? I'll call it PUR, right there on the chromosome. And let's just say this is a bacterial chromosome in which we have integrated one of those F plasmids. So now it's an HFR strain, right? Okay. And so this is contained in a cell. Okay, so remember that's the chromosome and this is the cell. And it conjugates with, right, a strain that has just a chromosome, right? But it also has a purine biosynthesis gene. All right, but this F- strain was picked out specifically and you didn't lose sound, guys. I took a few seconds to recharge my pen right there. This purine gene is present, but this F- strain was picked out specifically because there's a mutation in the purine gene that won't allow this organism to produce purines. Okay, it has to be supplied those nucleotides in the media in order to grow. All right, so let's just say we were to put some media out there. Okay, here's a petri dish, and here's another petri dish. And in this petri dish, we don't supply purines, so it's purine minus. And in this one, we do. Okay, well, this F- strain can grow on this petri dish, right, where purines have been supplied, but cannot grow on this one, right? So you'll see no growth, but you'll see colonies on this one, right? If you mix these together, right, and this chromosome gets transferred into the F- strain, right, let's just say the HFR strain is transferred and you get HFR piece, right, and you get part of the DNA, right? F- strain remains unchanged. It still can't grow on purines or without purines added to the media. All right, but at some point, right, in time, you're going to end up transferring that gene that is functional for purine biosynthesis, right? So let's just say now we have that gene has been transferred and we plate this F- strain on this plate that does have purine, so it can grow as it did before, but now without purine added, all right, you'll see colonies growing. Okay, and we can figure out what's that time, right, required for that purine biosynthesis gene to be transferred to the F- strain. Right? So now if we go back to this picture, here you can see a map of the bacterial chromosome, right? And on this bacterial chromosome, we have a whole bunch of genes. And so this chromosome has been mapped in minutes. This is 0 minutes, right? And then we go all the way around to about 100 minutes. Okay, so we've got 90 about here, and then we've got about 100 minutes. So the whole chromosome of this bacterium can be transferred into the new strain in 100 minutes, and each one of these genes were mapped. Here's a purine biosynthesis gene called PURB. There's a gene for histidine synthesis, that's an amino acid, glycine synthesis, another amino acid, arginine, all right, pyrimidine biosynthesis. So there's a lot of genes mapped, okay? And then if you sort of pan down, they've sort of given you a picture of the genes that are found between around 25 minutes and about 28.5 minutes. All right, so here are the genes that are found between those. So that those 4,000 genes in E. coli were mapped this way by finding many HFR strains and many mutants of E. coli that were deficient in these different biosynthetic pathways. And it took about 20 years, but we were able to map the E. coli chromosome long before sequencing was ever developed. Okay. Now, so we were just talking about plasmids having extra functions. These are independent of the chromosome. They replicate independent of the chromosome. They usually have a size that's much smaller than the chromosome. They range anywhere from 40 kilobases to about 100 kilobases in size. But even though these are extra functions or non-essential functions, sometimes they do carry some advantageous genes. Okay, they carry genes that allow them to do conjugation, that encode for the sex pilus and the replication mechanism of the plasmid that helps it be transferred into the new cell. They contain sometimes resistance markers for antibiotics, so that bacteria can live in the presence of certain substances, and it may be that they've lived in the presence of soil bacteria that are secreting toxic chemicals, and they are producing compounds to help them resist those chemicals. All right, so these can be contained on plasmids. They may have agents for destroying competing bacteria, so they may secrete antibiotics themselves or antifungals to destroy other organisms that are in the environment, competing for nutrients in space. And some of them, especially organisms that grow in the host, may be secrete toxins and things from the plasmid that aid in their pathogenicity, their ability to destroy host cells or attack the host in some way. All right, so we're going to talk about plasmids much later on, or a lot later on. But remember, these plasmids, there have been naturally occurring conjugative plasmids or fertility plasmids found out there that have as many as 5 different naturally occurring resistance markers, right? And these can be easily transferred from not only organisms that are closely related, but those that are not even related at all. So this becomes medically very important to control the movement of conjugative plasmid containing resistance markers from pathogen to pathogen. All right, so eukaryotes differ from prokaryotes in the way they package their genetic material. They usually have more than one chromosome. They're always linear. They have a nucleus, a true nucleus, so it's surrounded by 2 membranes. They package a lot more DNA than prokaryotes do, and they have much more elaborate packaging. Okay, so there, if we look at DNA of a eukaryote at the very highest level of magnification, there are globular proteins associated with the DNA called histones. Histones are these positively charged basic proteins that associate with negatively charged DNA. Remember, DNA has a lot of phosphate groups, and phosphate is a negatively charged functional group. And so that gives DNA an overall negative charge, and it electrostatically interacts with histones. Histones form octamers. They're small globular proteins, but they form octamers, and then the DNA will actually wrap around these octamers a couple of times, as you can see there. There tends to be a little spacer between these bundles of histones and DNA. By the way, those are called nucleosomes. So you'll have a nucleosome and then a linker and then another nucleosome. And in the EM down here, you can see a really high magnification. They look kind of like beads on a string. There are other proteins that associate with that DNA that package it a little more tightly. It brings the nucleosomes closer together in a structure that we call chromatin. And chromatin can actually be packaged either very, very loosely in the cell or more tightly in the cell. And so here you can see examples of tightly packaged chromatin. When it's tightly packed, it's called heterochromatin. Okay, so that's the tight packaging. And euchromatin describes when it's actively or loosely packed. Okay, so DNA that's actively being transcribed and used in a cell is in the form of euchromatin. Okay, and that DNA that's not being used at a given time, right, is packaged into what we call heterochromatin. So that's a tight packaging. Now, there are additional proteins that are expressed that package DNA in a highly tight form where there's no transcription going on, and the DNA is being moved from cell to cell in a structure called a chromosome. And you can see these under the light microscope in some organisms. So I'm going to answer the phone and end this video here.